DNA replication begins in the origins of replication. Proteins start the process by identifying the order of nucleotides and then attaching themselves to it. Then they separate the two strands, creating a replication bubble. The helicase then unzips the two strands of DNA, and the single-stranded binding proteins attaches themselves to keep the two strands apart. The parental strands are now available as template strands. The enzymes that synthesize DNA add nucleotides to the end of an already existing chain that is base paired with the template strand. This produces a short stretch of RNA. The primer, being the RNA chain, is synthesized by the primase enzyme. DNA polymerase catalyzes the synthesis of a new DNA by adding on nucleotides to an already existing chain. The DNA polymerase III will add DNA nucleotides to the growing end of the new DNA strand. Two new strands of DNA that are created are anti-parallel with their template strands. This anti-parallel repositioning allows the DNA strand that goes from 5 to 3 to be longer. This is where the leading strand's purpose comes in, synthesizing the leading strand. The DNA strand must be elongated in the 5 to 3 direction, working away from the replication fork. This is how the lagging strand is made. In the strand, there are a series of segments that are called the Osaki fragments. Polymerase III lays down the new DNA on the lagging strand, and the DNA polymerase I replaces the RNA primers with DNA. Then the DNA ligase attaches the sugar phosphate backbones of all the fragments into a continuous strand of DNA. The bond has to be strong enough to keep the newest strand of DNA and the DNA fragment together. Makes me feel fine. The DNA replication process continues until the strand is complete. The key point here is that the strands of the original and replicated DNA must be anti-parallel. Little light is shining through the window, lets me know everything's alright.